Put this microscopic bubble in a carefully prepared bath, and it can take in supplies, make proteins, copy its DNA, grow, and produce daughter compartments. Give one version an advantage, and it can outcompete another. That sounds alive, but nearly every step still hides a laboratory hand. This is SpudCell, a synthetic cell-like system assembled by researchers at the University of Minnesota. Its creators describe it as the first bottom-up synthetic cell to combine the major stages of a cell cycle. So did scientists build life, or an unusually capable imitation of it? The useful answer is not a yes or no. It is a map of what the system can do, what the laboratory does for it, and where the boundary still lies. First, the evidence status. The result was posted in July 2026 as a bioRxiv preprint titled, "A chemically defined synthetic cell capable of growth and replication." At the time of this production, it had not completed peer review or independent replication. The experiments were generally performed with 3 independently prepared samples, but that is not the same as another laboratory reproducing the whole system. The authors also disclosed a provisional patent. None of this makes the result false. It changes how confidently we should describe it. In this video, reported measurements are treated as findings of this preprint, not settled scientific consensus. Most famous synthetic cells begin with an organism that is already alive, then remove or replace parts of its genome. SpudCell takes the bottom-up route. The researchers start with a lipid membrane compartment and add purified biological machinery. That distinction matters, but the phrase built from non-living components can be misleading if it sounds like simple chemicals suddenly organized themselves. SpudCell contains ribosomes and many purified enzymes originally derived from bacteria. It uses a defined protein-making mixture called PURE, plus a DNA-copying enzyme from a virus that infects bacteria. The achievement is control and integration. The ingredients and their starting concentrations are known. It is not independence from existing biology. Inside the membrane is a genome of about 90.3 thousand DNA base pairs. Table S6 of the manuscript divides it among 7 plasmids. Together, they encode the DNA polymerase, an RNA polymerase, a membrane protein, a fluorescent reporter, and many parts of the protein-making system. The paper contains inconsistent prose counts in a few places, but its component table and measured plasmid list both support 7. DNA instructions alone are not enough. The starting compartment also receives working ribosomes, transfer RNAs, enzymes, amino acids, energy molecules, and DNA and RNA building blocks. A natural cell manufactures and recycles much of this machinery. SpudCell begins with much of it already supplied. To keep the reaction going, the researchers prepare smaller feeder liposomes. These carry fresh lipids for membrane growth and replenish enzymes, ribosomes, and other reaction components. SpudCell expresses a modified pore-forming membrane protein. A tag displayed on that protein can bind compatible lipids on the feeders, helping the membranes fuse. So the genome influences whether the compartment can receive supplies, and faster expression can mean more fusion. But this is not metabolism in the ordinary cellular sense. The laboratory manufactures the food packages, adds them at each round, and maintains an external solution containing the small molecules needed by the reaction. SpudCell can open the delivery. It cannot yet build the supply chain. The headline multi-generation experiment followed 5 rounds. In each round, the synthetic cells spent about 12 hours with feeder liposomes while proteins were expressed and DNA was copied. Then the researchers forced the liposomes through a membrane filter. That mechanical extrusion split and resized them. Fresh feeders began the next round. The team used molecular generation counters to show that at least some lineages received a feeder in every cycle. They detected newly made DNA, RNA messages, and fluorescent protein through the sequence. This is a substantial integration result. It is also 5 generations of mechanically assisted division, not 5 generations of autonomous, genetically controlled reproduction. Keeping that intervention visible changes the meaning of the claim. Copying a multi-part genome is only useful if daughter compartments inherit the pieces. After 5 mechanically divided generations, The researchers screened diluted samples and selected 95 putative single-cell aliquots for plasmid testing. The paper reports that about 30% tested positive for the complete set of 7 plasmids. That is both the impressive result and the central failure mode. Some compartments appear to have carried the entire genome through repeated feeding, copying, and division. Most of the selected measurements did not contain the full set. Without a cytoskeleton or chromosome segregation system, plasmids are distributed imperfectly. This is an approximate result from a screening procedure, not an unqualified census of every daughter cell. The researchers also tested a division mechanism linked to gene expression. This was a separate, single-cycle experiment from the 5-generation lineage. Proteins displayed on the membrane recruited externally added linkers and a binding protein. Crowding material on the surface deformed the membrane. In a bead-based assay, daughter compartments carrying DNA appeared only when the required division components were present. A stronger promoter produced more daughter-associated DNA marker. The team then combined feeder-driven growth with this division method and detected each genome plasmid in the pooled daughter fraction, not in every individual daughter. The method had lower yields and less uniform daughters, and it still needed linker molecules, the binding protein, beads, and laboratory handling. Genetically linked does not mean autonomous. SpudCell also connects a genetic difference to competitive success. The researchers deliberately prepared one population with the ordinary T7 promoter and another with a stronger T7 max promoter controlling the feeding-related membrane protein. The stronger version fused more efficiently. After 5 mechanically divided cycles, reporter measurements and sequencing showed that the stronger feeding population gained an advantage, especially when feeders were scarce. That is selection. A heritable difference affects which variant leaves more descendants. It was not open-ended Darwinian evolution. The useful variation did not arise spontaneously inside the cells. The researchers introduced it before competition began. The system can select among supplied variants, but it does not yet generate and explore variation as robust living populations do. So is SpudCell alive? Even its lead researcher says she does not think so, while acknowledging that life has no universal definition. The capability map is more informative. Protein expression, demonstrated with supplied machinery. DNA copying, demonstrated. Feeding and membrane growth, demonstrated with laboratory-made packages. Division, mechanically reliable across 5 rounds and genetically linked in a separate, lower-yield test. Selection, demonstrated with an introduced variant. Missing are self-made ribosomes, lipids and energy, reliable genome segregation, metabolic regeneration and regulation, the capacity for spontaneous evolution, and indefinite autonomous reproduction. SpudCell does not erase the line between chemistry and life. It makes that line easier to inspect, one dependency at a time. That is a more modest conclusion than creating life and a more useful scientific achievement.